And hello, everyone, for joining. It's my pleasure to introduce Dr. Ratan Rai, who is our today's seminar speaker. Ratan graduate using NMR from India. He has two postdocs in California and in Kentucky using NMR. And we're more than happy to have him as a portion of our fall service in the Chemical Genomics Core in the School of Medicine. And I can't wait to listen and to have you served by Ratan. So you have the floor, Ratan.
Thank you, Alan. Thank you for the nice introduction. And today topic of the seminar is structure-activity relationship by NMR. And we are going to talk about how to utilize fragment-based drug discovery approach for your projects. So let me introduce to you my core. So our core is a Chemical Genomics Core facility, and it is located in the Van Ness Medical Science Building, room number 105, 107, 1015A. And as mentioned, Dr. Alan Waffle is our co-director. And since 2016, a Chemical Genomics Core facility has provided more than 3,000 services to the faculty members from 48 departments or other academic units, and supported more than 135 grants, and out of which 67 are external. You can go on into the Indiana CTSI website and look for more information about our core services and policies. And the link is provided over here. So in our core, there are 4 people, and I myself is responsible for developing structural activity relationship by NMR in our newly acquired 600 solution NMR. And this will include developing and establishing the structural biology by NMR in core. Dr. Jingwei Meng is responsible to develop biochemical or cell-based assay screening with drug-like or diversity-based compound libraries. He also added a new addition to existing high-throughput technology, which is CRISPR screening to identify novel drug targets in the signaling pathway of your research interest. And he has also acquired a newly DNA-encoded library for high-throughput screening for drug designing. Dr. Zheng is responsible for chemoinformatics, biomedical chemistry, or analytical chemistry. His expertise covered design and synthesis of small molecule. He also expert in LC-QTOF and NMR analysis of small molecules, peptides, and and protein fragments. Dr. Waffle research focus on phase display libraries. His expertise including identify antibodies to the molecules of your interest. In our core, our facility is equipped with a Bruker 600 MHz solution NMR instrument with cryo-probe facility, and it has the auto-samplet facility also. So this process, NMR experiment process, can be automated, and we can handle large number of samples. We have a probe which can detect proton, carbon, and nitrogen, and we have also a facility to detect fluorine nuclei also. And this is going to be really beneficial for fragment-based drug discovery. So as a part of the services, these are the several services we provide in our core as a part of a structural activity relationship. So we provide protein structure determination in solution. We also help you to setting up fragment screening, and we are going to also identify your flexible region of protein, and we also do a verification of structural integrity of targets and protein construct. So if you have a protein and if you're looking for any kind of services from our core, we will help you setting up those services in your lab, and we can solve the structure of protein and help in to find a very small compound library which can be helpful for your fragment screening. So there are some pre-request arrangement if you wanted to do the protein structure determination using NMR, or if you wanted to have some screening by NMR. So the first requirement, which is very necessary, is you should have access to highly purified protein. And if you don't know how to make it, we will definitely help you with protocols and so that you can make in your own lab. And we have access to a couple of faculties over here who are regularly expressing those protein. So if you are facing any problems, we can help or guide your students to make your protein. And if you're looking to solve the structure of a protein, then another requirement is you are able to label those proteins. So we need required a minimum 15 and 13 in labeling so that we can solve the structure of a protein. And if your protein size is big, then we require additional deuterium labeling also. But if you're looking for only screening, then we don't require label protein. Unlabeled protein is really good for this screening protocol. And since we have only access over to 600 megahertz facility, then with this facility, we can solve the structure of protein up to the 30 kilodalton. And amount of protein we required is varied between 0.1 to 2.5 millimolar concentration. It depends. Some protein they are happy at even higher concentration, around 2 millimolar, and some proteins are not happy. So we can run those experiment on 100 micromolar. And because definitely if concentration is low, time will be a little bit higher. And another very important characteristic of protein which is required for structural determination is at least your protein should be stable for 2 to 4 weeks, because generally NMR is not a very sensitive technique, and so it requires a lot of time for data acquisition. And if you have any student who wanted to learn about NMR, then our core facility also give you and then our core facility also help you to train your students or graduate students, in which I'm going to teach about introduction to the NMR. I will teach them how to use Bruker software for biomolecular application. I also teach them how to set up various 2D, 1D, 2D, and 3D NMR experiments for their analysis, through which we can assign their backbone and sidechain assignment, and at the end they can calculate their own structure. And once the structure is known, then I can also teach them how to do the fragment-based drug design and or fragment screening. And these are the cost for use for these services. So suppose if you want training of your graduate or postdoc student, it costs generally around $800. For if you are looking for any kind of drug molecule and you want to see whether it's interacting to your protein or not. So for unlabeled or labeled protein, we are charging, if there is only one fragment, we will take only $25. But we have one library also, which can cost, if you are selectively picking them from our list, then it will cost something around $1,250. Definitely these costs we can discuss it while doing your project. And if you have some kind of data which you need to be analyzed and protein structure determination or for the backbone assignment, it will cost something around $3,000. And if you want the whole project, complete data acquisition and analysis, then it will cost you something around $7,500. So generally, if you look at the NMR spectroscopy is capable to provide information and answer to the biological problems that cannot be obtained by any other means. So it's very powerful technique, and it has application in around almost all the area of sciences. MRI is one of the popular application you must have seen in everywhere. And for all the chemists, NMR is one of the essential technique for small molecules. And definitely we can see some in-cell NMR also to track different kind of pathways, and we can do high throughput screening using NMR also, and it's more of the application is structural biology and drug discovery. So it can provide structure of a macromolecule in a different environment, ranging from buffered solution to intact cell. It can provide insight in dynamic processes and allow to monitor biomolecular interaction that are key to cellular responsible to an environmental, developmental, and growth signals. So, but in this talk here, I'm going to focus about NMR spectroscopy in drug design and discovery. So I realize that you may not know about NMR spectroscopy, so I will give you a little bit of a brief description. And if you have any question or any doubt or anything I show which you don't understand, please just ask question at any time.
Ratan, there's a question in the chat.
Yes, there is one question. So are all the atoms of Okay, so I'm gonna answer it right now in this next slide. So let's suppose that you have a protein, and why we need to uh, label it? Because in NMR, all the atoms are not detected through NMR. Why? Because proton is one of the most abundant molecule. So definitely we can detect proton and its isotope deuterium also. But one of the most common element, which is carbon, right? Carbon 12, it's NMR inactive nuclei. So we cannot detect carbon 12. So if you're looking for a protein and if you want to assign it, definitely we need to label as one of the isotope, which is carbon 13. So we need to label it. Similarly, for nitrogen 14 is spin 1 nuclei. So it's very difficult to get signal in NMR also. But if we label that protein with 15N, then we can detect signal from the protein. So if we are looking for the protein analysis, then we need to label it. But if you're not looking for the protein analysis, right, or structure of the protein or the dynamics of the protein, then you don't have to label it. So suppose if you want to screen your compound, whether your small drug is interacting with the protein or not, then you don't have to label it. But if you want to learn about the structure and the dynamics of the protein, how it moves, which has where is the binding pocket, definitely then we need to label it. That answers your question? So, like I explained in this slide that we cannot detect all the atoms. So what we do, so uh, we can grow our protein in isotopically labeled glucose, amino acid, or ammonium sulfate. And we will direct those specific labeled compound in such a way that our protein is well labeled. And usually we record a 2D spectra which correlate different nuclei in different ways. So this is the example of one of the uh, this is the example of one of the common experiment which is called HSQC, and this is called heteronuclear single quantum correlation experiment. In this experiment, we are going to see the signal from all the backbones. So how this experiment work? The magnetation is transferred from hydrogen to from hydrogen to attach nitrogen through the J coupling. And so for each of the nitrogen in the protein, we are going to see one peak. So suppose that your protein has 70 amino acids. So we are going to see 70 peaks over here. But there are some other peaks such as tryptophan side chain and asparagine and glutamine side chains over here. And tryptophan side chain is over here is also present in this spectra. And if your protein has proline, since it doesn't have an H over there, then we are not going to see the signals from proline. So in this particular residue, it has around 56 amino acids. So we will see around 56 peaks from each backbone amino acids, and some other peaks which are corresponding over here, which I put it through dot, or this tryptophan 57. These are the coming from the side chain. So once you have your label protein, then and if you want to solve the structure of the protein, then the target is to get the assignment. So what this term assignment means? It means this is your common backbone of your protein. So it in this example, it has alanine 17, serine 71, leucine 72. So in your protein, it can be different. So our main idea is to, based on different kind of NMR experiments, whether it's 2D, 3D, or 4D, assign each individual residue. And once the assignment is done, it will look like this. So we have assigned each amino acid, either this nitrogen, it is, or if methyl, or this CH or NH, we have assigned each individual residue. Once the assignment is done, then we will run a different kind of experiment called NOSI. NOSI is a kind of experiment which measures the distance between one proton to another proton through interaction called dipolar interactions. So with the help of those, we can estimate the distances, and these experiments called NOSI. So once you have a dipolar restraint, chemical structure, then we will use those constraints to solve the structure of a protein. And in NMR, you will not get one structure. You get an ensemble structure through which you will know that how your protein is moving and how many conformation of your protein is present. So this is one structure of a protein which are solved through NMR. You can see that which portion is moving very fast, which portion is moving very slow. So once we know that, we are ready for the structure-activity relation through NMR. So, how in NMR drug discovery process happens? So there are two methods. One is monitor NMR signal from protein in the presence of ligand. So suppose that there is a ligand and you want to see whether your ligand interact with protein, then we can label your protein sample and see the interaction. The other method is, suppose that you want to screen that you have 10 molecule, 20 molecule, or you want to screen a library. Then there are other methods in which you don't need to label your protein. You need unlabeled protein only, but it should be highly purified. So there are several methods. So I will give you a little bit of introduction how these methods works and how can be useful in your projects. So first of all, I'm going to discuss about in which protein is labeled and ligand is unlabeled. And this method is called and is structural activity relationship by NMR. So in 1996, Abbott Laboratory and Dr. Fisk, he presented one paper in the Science, and he discussed about this method. And how this method works, suppose that you don't have to look at for a very active compound. You can start with a small compound which has a low binding affinity of protein. It can be a very small fragment. And they wanted to do it to replace this particular molecule FK506, which is which was designed for as immunoprecipitant that binds to the FKBP protein. So in here, how it works, so they proposed a method in which protein is going to be labeled, and they say that let's find a small molecule which has a low binding affinity and try to find one particular region of a protein where it binds, and try to optimize it and try to find a small molecule, which has a binding affinity of micromolar. It should be high binding, and you cannot directly use as a drug. So optimize that first binding site. Once that binding site is optimized, try to find another small molecule, low molecular small molecule, and try to find a neighboring site. And it has some kind of binding affinity, and it has also low binding affinity over here, and try to optimize it so that you can find a two molecule with low binding affinity. Now, with the help of chemistry or any linker, we can synthesize a molecule which link these two molecules. So what will happen? You will find a molecule which has a higher binding affinity compared to these two places. So, and once you find your scaffold, you can build from there. So that's the starting point, that even a small molecule which has a low binding affinity, and with the help of NMR, you can find a first scaffold and you can build that scaffold. And from there, you can find your lead compound. How it works in NMR. So I showed you an HSQC spectra in the beginning, right? So this is one small compound, and it cannot be directly used as a drug. So how it's going to work? So this example is based on the BCLXL family of drug protein, and we wanted to see how this small molecule bind to this protein. So what we are going to do, we are going to titrate it. So protein is labeled and this ligand is unlabeled. So what will happen? We record different 2D NMR spectra in the absence and the presence of increasing concentration of compound 1. This is compound 1. So we find that there are several portions of the protein which doesn't move at all. But there are certain regions of the protein that moves very significantly. So one thing was clear, that its binding is selectively, it's not binding everywhere. And once we know which, because we have the assignment, so once we know which portion is binding, like glycine 94, glycine 138, and threonine 109, it's binding, then we can map all those resonances on the protein. So we can know that, okay, this is the one region of the protein which is interacting with our drug. Okay. So once we find that, okay, we can have a small molecule and this small molecule is interacting with our protein in certain region, then this method became really powerful tool. How it's going make a powerful tool? Because the ability to track changes in a protein due to small fragment or drug makes NMR a suitable choice for drug discovery. How? I'm going to show you in the next slide. So once we have one site is measured, like over here, then we try to find another site. And this is another small molecule which we find that. So in zoom spectra over here, you can find that to the molecule 1, there are several protein, glycine 94, glycine 138 moved a lot. And even in the presence of second uh, binding compound, which is fragment 11, we find that it moved glycine 94, glycine 138, but it also moved glycine 196. So we know that the second compound is binding in the vicinity of our protein. So what we will do, we will map, again we map this on this protein and we know that they are nearby. Once we know that two fragments on a protein have the two binding pocket and they are nearby, then with the use of chemistry, we can definitely combine them. So the first molecule is has the binding affinity of 300 micromolar and second molecule has a binding affinity of 6000 micromolar. And if we combine these two molecules through this linker, then we found that our combined bind affinity became 1.4 micromolar. So we take a jump around 100 folds by doing a very simple experiment. And from there, we built this molecule, which has a binding affinity of 36 nanomolar, and further modification of this molecule end up into the drug, which is called Ventoclax, and it's a drug which was approved by the FDA, and this is used to treat the chronic lymphatic leukemia. So one small experiment, and without to go through a very huge library, you choose a very small fragment, and those fragment end into a very nice drug. So this is the one way to look at it when your protein is labeled. Let's see when your protein is not labeled, how is NMR going to help us to design different kind of drugs. So there are several experiments we can utilize it to screen different compounds. One is T2 or T1 row relaxation. Another is saturation transfer relaxation. Another is Waterlogzy experiment. One is diffusion editing experiments, and another is 19F-NMR screening. So I'm going to talk a little more about these methods. So let's see, what do you mean by ligand by T2 changes, T2 NMR or relaxation edited experiment? So in NMR time scale, our drug is really small. So it move really fast, it tumble really fast. So we will get very sharp line. And with this, if this small molecules bind to some protein, then what will happen? Its movement will be restricted because it's binding to the protein. So once its movement is going to restricted, then the property relaxation T2 is a property of NMR. It's going to be decreased. So our line width is going to be increased. So this is one example of a molecule in which they found that they have used a mixture of 10 molecule, and once they interacted with our molecule, then line was brought in out. And so it can be used to screen maybe thousands of compound using this method. Another method is ligand-based method, which is called saturation transfer difference experiment. In this experiment, we are going to irradiate our protein with RF frequency. So once it's going to be saturated, then if protein is saturated, then it will be transferred to the drug. Suppose that, let's see, in a free NMR pool, there are 10 drug present in NMR tube. So what will happen? If our drug is binding to the protein, then from this saturation, RF frequency will transfer to our small molecule, and we will see change in the intensity pattern. And if we record two experiments, one off resonance, one on resonance, then if there will be interaction present, suppose there are pool of 10 molecules and it's interacting with one, then what will happen? If we subtract one from two, then we will get a difference. Rest of things will be zero. So in this particular, there are example, there are 4 molecules, one is interacting. So in this case, we will see a positive signals from only one molecule. Rest of things which are not interacting, we will not see any signals. So this is the example of human serum albumin in which we have put tryptophan and arginine. Tryptophan interact with human albumin, but arginine didn't interact with it. So we will get in different spectrum, we will get spectra from tryptophan only. There will be no arginine. Another popular experiment is called water log Z. It is one of the popular experiment people use for different drug screening. So water log Z experiment works on the transfer of water molecule to the drug, and this transfer can be done in different mechanism. It can happen that our water molecule is directly transferred to our drug, or it can happen that suppose that there is a binding pocket and there is drug, drug is binding in that binding pocket. So what will happen? It will repress all the waters from here, and that is the surface, because this is the surface of the protein and there are so many water molecules, so it will interact through this pathway. Another pathway, it can happen through chemical exchange, because there are several NH resonance through which it can interact. So suppose that they are a mixture of 20 compounds and we do water log Z. So what will happen? If our compound is binding to our drug, our compound is binding to our protein, then we will see a positive signals. Rest of them will show a negative signals. So it can be useful in screening around 100 or even thousands of molecules. Another experiment people use it for high throughput screening is called diffusion editing experiment. This experiment is designed in such a way that it can separate out a different molecule based on their size. Suppose that you have a mixture of 20 compound, then based on their size, we can easily tell them which molecule is this. So this is the one simple spectra in which there are 4 molecules are mixed together. Based on their diffusion constant, we can separate out these 4 molecules. And how this method work for different drug interaction. So suppose that you have a mixture of compound, and then you will record a dosy experiment. Then we will add a protein in it. So if our protein is interacting with our drug, then it has a different size. So definitely the lines will be different. So if we subtract a spectra 1 to B, then we will find all those molecules which interact with our drug. So this is also a very rigorous method to screen different kind of compounds. So another new method or service which are presenting for our core with the help of CDD, and we bought one library, and this library was bought with the help of cancer center, and we bought a library of around 1200 compounds, and which has fluorine label. So what do you mean by fluorine label? It means one of your site has a fluorine over here. Like this is compound 1, it has fluorine over here. This has another example in which fluorine has a here. So either it attached to one of the end. And why the fluorine is such a excellent nuclei for drug discovery? Because in this whole molecule, if you look at the proton and NMR spectra, we will see around 15 to 20 signals. But if you look at the fluorine, since it has a one site, we will see only one line. So this is one example through which we are going to show that because fluorine has a very wide chemical shift dispersion. So what it can happen that if you like to test 20 or 30 molecule one at a time, we can do it. So this is the range, like -50 to -230. So generally CH3 come around this particular region, CH2 come around this particular region, and CH come around this particular region, depending on what kind of group is attached over here. And how we see the interaction is happening or not. So this is one example which has recorded at 600 megahertz NMR, and the blue line is with without protein. It is a mixture of 20 compounds. So there are 20 compounds are present in this mixture. And when we added protein, we can see that out of 20, 18 show no difference. But out of 20, one show a very drastic drastic change in signal intensity. So this is the clear hit. And second one, it show a little bit. It means at that particular concentration, it has showing some interaction. So you in one go, you get a clear hit and one potential hit. And these changes can be a little bit, you can get a chemical sheet perturbation also, or it might happen when you added a protein that fluorine signal completely go away, which indicate very really tight binding, and it can show a different shape or a chemical sheet perturbation at all. So this is the example in which there are 50 molecules are put into one NMR tube and try to see the interaction. So one of the good thing about having a fluorine mediated library, that simplicity of this analysis is quite easy. So you will see only couple of residue, and based on those residue, either they have a change in intensity or chemical shift perturbation. And you can clearly indicate that, okay, this is our compound which interact with our protein, and it can be a hit. And from those hit, you can make your potential lead compound. And that's it from my side. And let me know if you have any questions.
I had a couple questions. So the first one, just kind of about the last part where you talk about the fluorine marking. I've seen in a couple different papers where they use a fluorine atom to basically change the electron density of particular molecules.
Mm-hmm.
Do you see any sort of effects in that realm on using fluorine-based fragment screenings?
Not really. So generally we are looking at change in the fluorine environment, right? If it either is interacting with your protein or not. Suppose that there are a mixture of 10 compounds, and you are looking, because we are looking at the particular interaction, it's binding or not. If it is not binding and it's minding its own business, then you will not see any interaction with the protein. Definitely negativity will increase. And if you have, suppose you have a protein which has electronegativity, completely electronegative protein, that it might hamper it. But I have one experience with one of the screen which I am doing with Professor Millie Georgiadis, and it is a DNA binding protein, and one of those side is quite electronegative, but still I'm not seeing any interference from this particular library.
Hmm. Great. A second question I had is so there the computational side of the business seems to be really hot with machine learning based generative fragment construction.
Yeah.
What are your thoughts on that? I know, I mean, of course, everything you've presented today is really good for like the true validation, right? Like you need to do the actual biochemical screening to say something with confidence, whereas a lot of in silico or like generative AI docking type of things can be a little bit hallucinogenic, is the word that I've heard them use. But do you have any hopes to see any sort of anything in the field on that side that can help to sort of aid or perhaps even like compete with like the true fragment screening with NMR?
Yes. So even what we are doing over here, right? So we are trying to find one molecule, but definitely look, in silico modeling is really good, and you can screen even millions of compounds, right? It's quite easily you can go there and screen it. But definitely you need some biophysical proof and how it interact in actual scenario. Because sometimes it do happen that what kind of structure you start with when you are screening, whether it's NMR structure, whether it is the cryo structure, or whether it's the X-ray structure, you might find that because if it is not NMR structure, the flexibility is not there. So you might find a really good hit through in silico modeling, but if you look at in real biophysical technique, it won't be as good as it seems into in silico modeling. But definitely it helps because it will give you some kind of binding pocket where it binds, and even if we find a hit over here, we definitely go back and see in silico modeling how it works, because our idea is, because these are the small fragments, these are not drug. So idea is, if we find one pocket and try to see where it binds, and we have some kind of modeling over there. Suppose that your protein is not, structure is not solved through NMR, and it has a cryo-EM or X-ray structure. Then in that case, those in silico modeling is really important, because there will be no way to look at where it binds. But in silico model, we can confirm, okay, this is the one binding site, and this, and for the other, this is the second binding site. From there, we can have some kind of chemistry involved over there, because it's a complementary. There will be no replacement. But yeah, it's going to, I will say, this in silico modeling and the NMR is going to be complemented to each other when you don't have any structure. Suppose that you don't have to solve the structure of a protein, then at least you should have something to look at.
Yeah, so I guess kind of related to that is uh what do you think are the biggest limitations on using the in silico side? You mentioned flexibility as being one of those, and of course having the correct structure to start with, I think is of course pivotal, right? Can you sort of elaborate on what else you think the in silico side, what the field needs to address to get to the level where it could compete with like a hard biochemical assay like NMR?
I think the quality of a structure is going to change. If you have a really good quality of a structure, and you know which uh, portion of protein is flexible, which is dynamic, which is rigid, that will help in silico modeling. Because suppose that if you have a crystal structure with maybe 3 angstrom, and you find a binding pocket which electron density is not well mapped, then it might looks really good in silico, but when you do biophysical, it will crash. So as soon as you improve the quality of a structure, in silico modeling is going to do a really good job.
Great, thank you. And I found your talk very insightful. It's really, I found it previously kind of hard to understand a lot of the chemical shift plots, and I think the way that you described them was very helpful. Thank you.
Okay.
Hey, Ratan, it's Mark Kelly, but let's face it, nothing beats good data with real molecules over in silico, right?
Yeah, that's true. That's true. So like for For one, like for ape1, like we find one out of 200, we have done up until 200. Up to 200, we find one hit. So, and we proved through different experiment that it's a real hit. So will be no replacement.
Right, yeah. Once you have something real like that, you can go from
Yeah.
there. So, yeah. Anyway, thanks. That was very nice. Good explanation. Thank you.
Thank you.